Presentaré a los ponentes de esta mañana, and I will switch to English to introduce Dr. Brad Jones. He's a viral immunologist an associate professor at the Well Corner Medicine Center at the Division of Infectious Disease in New York City since 2018. He received a PhD in immunology from the University of Toronto, and then he was doing his postdoc at the Reagan Institute, MH8, MIT, and Harvard in 2012. He is a principal investigator of the research enterprise to advance the cure for HIV, REACH, one of the NIH flagships, Martin Delaney collaboratives. And he has put a lot of effort over the years to increase and to understand how cytotoxic T cells and the immune system can cure HIV infection. So welcome, Dr. Jones, and you can start. Thank you.
Great. Thank you so much, and good morning, everyone. Thanks for getting up early. So lovely to be here and to reconnect with some old friends and meet some new friends and learn some about the great science happening in Spain. So I'm going to talk today about a mechanism by which HIV may persist in people on long-term therapy by perhaps being seen by cytotoxic T cells, but not succumbing to CTL killing very readily. So first we'll talk a bit about HIV-specific CD8 cells in general. So we know that CD8 T cell responses play an important role in controlling HIV replication. There's three kind of pillars of evidence for this. First of all, the CD8 T cell response comes up at around the same time that viral load decreases from a peak to a set point. Rare individuals called elite controllers, this is associated with certain HLA-B alleles, which implicates a key role of CD8 T cells. And in non-human primates, if you deplete CD8 T cells, this causes a spike in SIV viral loads. But it's also complicated. And the ability of CD8 T cells to control HIV varies depending on each individual. And I know it's a bit risky to try to use football metaphors as a North American being here in Europe, but it's early in the morning, so I thought I'd try to make it fun, and hopefully I don't mess it up too badly. All right, so the HIV-specific T cell performs really well in some settings, like in elite controllers. In other individuals, it still kind of does its best, and it has an impact, but maybe not to the same degree. Just like some players might perform better in Barcelona, for example. So why can't CD8 T cells eliminate HIV after ARV suppression? So you come in, you give these very effective drugs, you've shut down the virus. So in the football analogy, the other team's not playing anymore. Why can't the CD8 T cells finish the job and eliminate those last remaining cells? And so that, in my ongoing metaphor, is a bit like missing a penalty kick here. It should be relatively easy now to get rid of the cells that remain. And the classic explanation for this is that the reservoir is latent. It's hiding as a provirus here in the infected cell, but it's not expressing any proteins, it's not making any antigens, and the T cells can't see it. So the T cells are trying to score the goal of killing the infected cells blindly because they just can't see the infected cell. And that's, of course, a very important part of why HIV persists, but my talk today is going to try to convince you that it's not the only way that HIV evades cytotoxic T cell responses. And there's a number of lines of evidence here for the idea that CD8 T cells continue to see some infected cells in people on therapy. And if we can understand why those aren't being killed, maybe that can open up some exciting new therapeutics. And that's going to be kind of where we end the talk, is on new therapeutic potentials. So these are data that comes from Matthias Lichterfeld's group, published, I think it was last year. And they used a new method to look at the proportion of cells with integrated intact proviruses that were transcribing HIV RNA. And you can see that although this does decline over time, there's still appreciable numbers of cells that have HIV RNA associated with them, or even high levels of HIV RNA. So these don't really look like they're in this latent state that's hiding from immune elimination. And from our work, we can ask the CD8 T cells directly in a way, are you seeing HIV-infected cells in vivo? And the way that we do this is we look at whether the kinetics of the CD8 response have any correlations with the HIV reservoir measurements, and we find that they do. And also, in particular, T cells that recognize early gene products like NEF maintain sometimes an effector phenotype. They're ready to produce granzyme B over short-term in vitro stimulations. And that's really a hallmark of a T cell that's seen its antigen recently, not 10 years ago when antiretroviral therapy was initiated. So I think it's becoming more appreciated that there is some interplay. So why aren't at least these transcriptionally active cells not eliminated? And first, I'll acknowledge one possibility, one simple possibility. Maybe they are getting killed to some extent, and maybe they're being replaced. And that's no doubt part of the truth. But I also want to switch here now and talk about this idea that maybe some of them have also been selected for cells that are intrinsically hard to kill, even if they're seen by CD8 T cells. And so these are two more slides contributed by Matthias Lichterfeld's group. And this is their newest sequencing platform. And to just encapsulate this in a kind of short summary, essentially what they're able to do is capture single-cell data of almost full-length HIV genomes along with surface phenotypes. So they have tags to a number of antibodies on the surface of those cells. So now they can ask for the first time, what is the phenotype of the cells that have HIV proviruses in people living with antiretroviral therapy without having to reactivate those cells first? And what they see, and this study is now in press, it's not out just yet, one of the things that they see that was interesting to us is overexpression of some negative immune regulators. So a little bit PD-L1, and more this CD155 or PVR, and especially if you look across here, these are the cells infected with intact HIV proviruses that can make antigen, and these are the ones that have intact proviruses and have undergone clonal expansion. We know that some of these defective proviruses can also make antigen, but this selection is really greatest with these intact proviruses. And so this is true. I'm going to focus a little bit on CD155, but they see this also for HLA-E and HVEM. So Matthias has called these don't kill me signals, essentially, because these can engage with inhibitory receptors on the CD8 T cell and dampen the signaling and protect that cell, perhaps, from being effectively killed, even if it's expressing HIV antigens. So kind of switching back to our work now, this ties into a hypothesis that we put forward quite a while ago now that proposed that one of the ways that these infected cells persist is they have been selected for cells that are resistant to being killed by CD8 T cells. And our main observation for this, and it's published work, so I'm not going to go through all of the data, but essentially we took ex vivo CD4 cells from people on therapy. We exposed them to our strongest shock and kill agents, so strong latency reversing agents, strong HIV-specific T cell clones, and we were surprised to see that we did not measurably reduce the reservoir by QVOA. However, we could take the virus that grew out of these QVOA wells, these viral outgrowth wells, infect new CD4 cells, and the same CTL clones could kill them just fine. So we started thinking, is there something special about these cells themselves that have survived for years or decades in people on long-term therapy? So I'm going to propose, before getting into some of the newer data, that perhaps there's a paradigm shift in progress, or maybe probably both parts of this are really important. The classical thinking is the reservoir hides from HIV, and that's no doubt true to an important extent. But there's also transcriptionally active infected cells that are apparently able to proliferate and persist, which may also be an important barrier to cure. So I've been talking about this idea that some cells may be hard to kill. Is there any precedent for that in other settings? And it turns out that this is something that's really well known in the setting of immuno-oncology, especially as checkpoint inhibitors start to work. And what I'm trying to show here are two movies. The first is a CTL engaging with a target cell, and it kills it very effectively. The propyridine iodide shows dead and damaged cells. Second one, which I seem to be having a bit of trouble with, shows two cytotoxic T cells attacking a melanoma cell. And here you can see there's a blush of propydium iodide. The cell's getting a bit damaged, right? The CTLs are clearly attacking, but it's able to resist this attack. And this has been, as I said, there's a wealth, it's all pretty recent literature, but there's a wealth of literature showing that cancer cells acquire this ability to resist attack, even if they're being recognized by cytotoxic T cells. And this was really summed up in a beautiful summary. If you're interested in the work I'm talking about today, this is not my review, but I strongly encourage you to read this beautiful review of ways in which cancer cells can resist CTL attack. And this assumes that the cancer is immunogenic. So we're not talking about does the cancer express antigen or not, is an infected cell latent or not. We're saying the infected cell is being recognized, the CTL is trying to kill it, and the cancer cell is resisting it. And there's some rapid defense here. So some of the rapid defense mechanisms, some tumor cells are able to rapidly secrete into the synapse proteins like cathepsins that chop up the perforin that's coming from the cytotoxic T cell and trying to punch a hole in it. So it can actively defend by using a protease against a protease. We see really dynamic actin remodeling in two kinds of ways. So the cytotoxic T cell has to exert a force on the target cell in order for perforin to be effective against it. And so target cells can adapt by being squishy, for lack of a better word, so that they can be deformed by the force and they're not as subject to killing. They can also put up essentially walls of actin, and you see actin accumulating and proliferating at the site of attack. So that's rapid defense. Slow defense, you can get accumulation of damage after a CTL attacks a cell. It can damage the organelles like the mitochondria, and autophagy is the process by which a cell takes up and eats its own damaged organelles. So a cell can just try to keep up with the damage that a cytotoxic T cell is doing. And if it can do that, it can live to survive a CTL attack. And especially in cancer, you see some mutation and dysregulation of regulated cell death pathways like apoptosis, membranes that can change over more quickly, etc. And so one example here would be overexpression of something like BCL2, which is a part of the apoptosis pathway. And if you have high levels of BCL2, that might make you harder to kill. So don't worry about the complexity here. The complexity is the point. We don't need to look at the details. The point I want to make is this is not a cytotoxic T cell. This is a target cell. And this is a snapshot of the complexity that happens within a target cell that receives a signal to die. So if this was an HIV-infected cell, it would already have expressed its HIV antigen, been recognized by a T cell, received perforin or granzyme or a fast ligand. That doesn't necessarily mean the cell is going to die. For example, you have these pro-survival factors like XIAPs, which can block the caspase cascade. And you can have overexpression of BCL2, which can block other steps of death. So the target cell is not a victim of the CTL attack for the most part. It's an active participant in this process of deciding to die once it receives a signal saying that it might be infected by a virus. One more complex slide, because I've never found one slide that captures all of the different defense mechanisms that's been shown in tumors. This is what I talked about earlier, release of cathepsins into the synapse, which can degrade the perforins coming from the cytotoxic T cell, autophagy to remove damaged organelles. Serpin B9 is another granzyme that degrades, in this case, granzyme B. Cell stiffness makes it harder for perforin pores to form. The lipid order and composition has an impact on how well perforin can form pores in the target cell. There's a lot that the target cell has to say about whether or not it dies. Okay, so that's all in melanoma. Does this have anything to do with HIV? So to ask this question, we set up an assay that we call a sequential killing assay. So these are primary CD4 cells. They're infected with either a wild-type HIV that can be recognized by a cytotoxic T cell clone, or an escape mutant HIV that has a mutation that means the CTL clone can't recognize it. So targets for killing, non-targets for killing. We label them with different colors, we put them in the same environment, and we try to kill them with a CTL clone that only recognizes this one population of cells. After this, we remove this cytotoxic T cell clone, And then we come in with a second clone that recognizes both viruses. So now everything's a target. And so one of two things is essentially going to happen here. In the first round, you're going to get killing of the wild type cells and not the escape mutant. And in the second round, they're both going to be equally killed again, right? Or maybe you'll have killing of the susceptible ones the first time. The ones that survive are resistant. And so in the second round, you only get killing of the ones that weren't selected the first time. So I hope that makes sense. But that would really tell us directly, is there such a thing as CTL resistance? And that is what we see. So you can kind of focus here. So this is one round of killing. We've killed off many of the cells that are targets, that have the wild type epitope with this clone. We come in with a second round of killing with a different clone, and we see now we've killed off a lot of these, but we haven't finished off this small population of cells that resisted this first round of killing, even though they're all sitting there in the exact same well, in the exact same environment, the same effector to target ratio. So we can also see this in a movie. So if we could start a movie on this, please. So this is an HIV-infected cell in green. Oh, sorry, I didn't know that sound. So you can turn this out if you want. But yeah, so here's the cell expressing HIV. It's a green fluorescent protein. The blue is a cytotoxic T cell. Just as in the melanoma, clearly the CTL is interested. It's trying to kill the target cell. We know it has cytotoxic properties. And yet this cell resists. And this was a cell that came directly out of that sequential killing assay that I showed you earlier that had survived this first round. So we've done many experiments like this, and I feel very confident saying that there are rare populations of HIV-infected CD4 cells, even from in vitro infections, that are resistant to being killed by cytotoxic T cells. So the question is, do these preferentially go on to form the HIV reservoir? And are these maybe selected over time in the HIV reservoir now that we know there's these complicated clonal expansion dynamics that continue to happen? So we want to then use a very similar system to ask what are the mechanisms of resistance. I showed you a whole bunch of mechanisms that are known in the setting of melanoma, but what about a primary CD4 cell, which is going to be quite different? So similar setup, we have our targets and our non-targets. They're both infected with HIV, but they have that one point mutation that makes these ones not targeted by the T cell. We mix them together. We expose them to killing by a cytotoxic T cell. We kill off most of these targets, and we have these bystanders to compare them to. And then we do some omics. We do some bulk RNA sequencing and some single cell work that I'll show you just a little bit of. So for flow aficionados, you can look at the details here. For others, I'll just point out the main points. These are results if we don't have any cytotoxic T cells. Here's replicates if we added cytotoxic T cells. And the first thing to see is these are the ratios of targets to non-targets. So once we've added our CTL clones, we've skewed these ratios quite a bit. We've killed a lot of the target cells. And then if you gate on these, and you look at the ones that are expressing HIV envelope, we're using two different antibodies, you can see even a further reduction. So we've really killed off most of the cells that express this wild type virus, but there is this survivor population that we want to know more about. And then these are coming from the exact same tissue culture well, they're all HIV infected, they just weren't targeted because they were escaped. So we're not looking at latent versus active infection, everything here is active infection. It's just a question of cells that underwent CTL selection versus cells that were not subjected to CTL selection. So the transcriptional profile tells us that there is definitely a difference between these populations. These are 4 different donors. Experiments were done on different days, so this is a very robust signal actually. And these are the gene expression profiles of the targets versus the bystanders. So we can see there's a lot of differential expressed genes. Over here, what we see in the PCA plot, this is if we don't add any CTL clone. So there's not differences between this escape mutant and wild type virus infection itself. But once we apply the CTL clone pressure, we can see this difference emerges because some properties allow those infected cells to survive. So there's a lot for us to dig through here. We got a lot stronger signal than we expected, so I'll highlight a few things. First, for some reason that we don't really understand, there's higher levels of interferon signaling in cells that survived killing. these cells are also seem to be deficient in cytoskeleton and junction formation, so that makes a lot of sense. They're less able perhaps to participate in the immune synapse with a cytotoxic T cell, which might make them harder to kill. And they have less expression of ribosome and translation machinery, which at this point we also don't know exactly what that means. If anyone has any ideas, let me know. And just to focus in on a few of these, again, underexpression of genes involved in actin cytoskeleton. I told you earlier about how physical properties can affect susceptibility to killing. So we're exploring, and we have some preliminary data that these cells may be just squishier, and squishier cells are harder for cytotoxic T cells to exert force on and to kill. And to highlight here also cholesterol homeostasis, I mentioned the lipid composition is important for perforin to attach to a target cell. And this has certainly been seen also in the cancer setting, where if you can actually deplete cholesterol with different drugs, you can make target cells intrinsically more susceptible to killing. So this is an example of one of the other crossovers from the cancer field that we are exploring as a potential therapeutic approach to make more cells susceptible to killing. So I'm just going to show a little bit of data here. This is the same experiment, but this is single-cell RNA sequencing, and it's actually site sequencing, so we also get some surface protein expression profiles. And again, you see with the clusters, the survivors cluster really pretty differently from the bystanders, so they're transcriptionally very different. And we were interested in this one gene in particular, CD155, because I mentioned earlier, this is one of the genes that was overexpressed in the real reservoir harboring cells. So something that might be really protecting the reservoir from being killed. And we see indeed in our experiment, these are all single cells. The cells that survived killing tended to have quite a bit higher expression of this CD155, which can interact with inhibitory receptors and prevent the CTL from killing and doing its job. And there is an interesting poster talk earlier at this conference too, showing that actually blocking this axis can enhance T cell immunity and HIV specific degranulation. So this might be a target that is, I think it's a target that's definitely worth exploring a bit more. So there's a lot for us to parse through. We to be honest, didn't expect to see so many differences between the survivors and the bystanders. I'm going to take you through in the last 9 minutes or so one of the stories that we've kind of completed, which is looking at one of the pro-survival factors that we also saw was upregulated in cells that survived this CTL killing called BCL2. So we're doing these in vitro systems. We're seeing that a lot of genes are differentially expressed in cells that survive killing, but we always have to tie this back to the real HIV reservoir, these cells from people who have been on therapy. So BCL2 high cells survive preferentially in vivo. They also are enriched for the HIV reservoir in cells from people on antiretroviral therapy. So you take cells from people, you sort into BCL2 high or BCL2 low cells, and you find that more of the HIV reservoir is present in these BCL2 high cells. That's in total CD4 cells. You can break that down more by the maturational phenotype, and you see that within the different maturational phenotypes, we still see this skewing. So there appears to be some evidence, it's not necessarily evidence for selection for BCL2 high cells, but the reservoir is preferentially hanging out at least in these BCL2 high cells. So this led us to ask if we could overcome this resistance, because there's an approved drug called venetoclax, which is a BCL2 antagonist. And these were the first experiments that I told you about from the 2018 paper, where we first thought of this idea of CTL resistance, because we were combining strong latency reversing agents and CD8 T cells and measuring the HIV reservoir and not seeing any difference, really to our surprise at the time, because we thought we were hitting these really hard. But finally, when we add this BCL2 inhibitor, we were able to see some consistent reductions here, even though they still vary amongst people, and there is quite a bit of bystander toxicity that you have to be very careful to account for with this BCL2 inhibitor. So instead of the CTL being blind to the infected cells, maybe it more so faces a wall of resistance here, and we're just starting to discover what these different factors might be. We know there's overexpression of BCL2 and pro-survival factors. Squishy cells are hard to kill, so low cellular rigidity, resistance to forming synapses. We had that really strong cytoskeleton and cell synapse signal from our transcriptional profiling, interferon gamma signaling, and others. So what does this mean for potential treatments for people with HIV? Well, we hope we're building a foundation from which we can identify therapeutic leads for our clinician colleagues and friends to start to test. And I'm happy to say that this has started to go forward now with venetoclax, which I mentioned is a licensed drug to treat leukemia. So there's a fair bit of clinical experience with venetoclax, quite a lot of clinical experience, actually. It's licensed for the treatment of CLL, SLL, and also AML. And there's information on the dosing for you there, and also the adverse events. So, you know, manageable, not trivial, but I would say manageable, and at least the investigators in this study believe reasonable to test in otherwise healthy people living with HIV. So Thomas Rasmussen at Aarhus University is doing this study, and he provided me with this slide. So they're in the process of doing a dose escalation study currently, just to look at tolerability in this cohort of antiretroviral therapy treated people with HIV. And then they're going to identify the maximum tolerated dose and move on to a larger study to see whether this results in any impact on the HIV reservoir, and also simultaneously looking at any impact on HIV-specific T cell function. So this study does not include at this point anything to enhance the CD8 function itself. Our data would suggest that maybe a combination of things that enhance CD8 function and overcome this one mechanism of resistance might be beneficial. But it's nice to see this starting, and I think our data point to a number of other leads that could follow in this footsteps, hopefully fairly soon. So how do we contextualize this? I'm talking a lot about resistance to killing. We know that HIV latency is a major mechanism of persistence and something that enables cells to hide from the immune system. And so sometimes when I present these data, I hear people look at it kind of pessimistically. So, oh great, now even if we overcome latency, which is already hard enough, we have to deal with resistance to killing. And I would look at it a bit differently. I think we know that there's a spectrum in the reservoir in terms of there being quite an active reservoir and a deep latency reservoir. And recent studies have shown that some of these deeply latent reservoirs can probably never reactivate. So what I would say is that let's see if using things like BCL2 antagonists, cell stiffening agents, CD155 blockades, I didn't talk about EZH2 inhibitors, if we can make a dent in this reservoir early, and then perhaps we have to combine with latency reversing agents to kind of shift our way down here into more of the latent reservoir. But to go back to Matthias's figure, can we come up with an intervention that we can give to people on antiretroviral therapy that at least knocks this out? You know, instead of having this slow decay of transcriptionally active cells over years and years of art, can we accelerate this by enabling the cytotoxic T cells to do their job better? And I think if so, it would be a win for the cytotoxic T cells. So last slide, just to try to put this all into a bit of context. The HIV reservoir is complicated and multidimensional, and this is a figure that we put together to try to encapsulate all of these factors, including CTL resistance, which I talked to you about today. So we know that infected cells are present in many tissues, as well as in the blood throughout the body, primarily in CD4 T cells and resting CD4 T cells, but also in cells of myeloid lineages. These cells exist on a spectrum of resting to activated, so there are resting cells that are latently infected, but there are also cells that are more activated if you look in someone who's on antiretroviral therapy. So Some of these cells are resistant to being killed by immune responses. That's probably more important for this activated part of the reservoir, and maybe somewhere we can have an impact with these drugs that can overcome blocks to resistance. And then if we zoom, this is less pertinent to today's talk in particular, but if we zoom way in, and there was some nice talks about this yesterday as well, we need to also think about where the HIV provirus is integrated within the genome of a given cell. It's not that we have to kill every cell that has an HIV provirus to cure someone of HIV. We just have to kill every cell with a provirus that is likely to reactivate. And those might be two quite different things if you have proviruses that are inserted in gene deserts, centromeric regions, and other transcriptionally quiescent places. And the thought I want to leave you with is that what really excites me about studying this area, in addition to the fact that cytotoxic T cells are intrinsically cool, and it's amazing how many different ways they have of killing a target cell, is that it potentially opens up a whole new array of different things to test in the setting of HIV cure that we perhaps hadn't thought of in this setting before, but which are more advanced in immuno-oncology. And BCL2 antagonists may be kind of the vanguard of this, but I mentioned briefly EZH2 inhibitors, BCL-XL antagonists. Hopefully we'll have a lot more leads coming out of our pipeline. So with that, I want to wrap up and thank a very talented graduate student, Louise Lair, who's led a lot of this work, along with Alberto Herrera and Paul Zumbo, who supported the single-cell data. Jared Wheeler is a technician who works very closely with Louise. A few people provided me with slides, which were very helpful. Thanks to my other lab members, the NIH for funding, and acknowledging our Martin Delaney Collaboratory that supports much of this work. Thank you so much for your attention.
So we will have some questions. Mary-Jay?
Thank you, Brad, for this wonderful talk. I have a couple of questions. The first question is, you think the cells that end up being resistant have a particular phenotype to begin, and that's why they end up being resistant? Because at the end also, the fact that they are being resistant to a CD8, is that physiological? Is the HIV, it's something that the HIV is doing, or CD8s are doing to the CD4? So what's your thought on that?
Yeah, that's such an important question. And I think it's probably a mix of both, but I don't have the data just yet. What I can tell you is if we do the same types of experiments I showed, but we use peptide pulsing instead of HIV infection to make the cells targets, we do still see some resistant cells. So there are some resistant cells even without HIV. There is a natural heterogeneity in CD4 cells. What I don't know yet is if the transcriptional profiles of those looks the same as that of the HIV-infected cells. So I imagine there are times where HIV is lucky and lands in a cell that's intrinsically hard to kill, and that cell then has a fitness advantage in terms of going on to form the reservoir. But in many other times, it's probably a cooperation with HIV. There's probably viral factors that may interact with host factors in some way that confers this advantage. And one thing I can point to there is, I mentioned EZH2 a few times. So EZH2 is a histone methyltransferase, and one thing it does is reduces expression of MHC1 by some degree. And so that might be cooperating with NEF-mediated MHC1 downregulation to protect the cell. So I think it's probably a combination of both.
But do you think they have a particular phenotype before?
Yeah, a particular phenotype that you can pick out. So what I can tell you is we've looked at this at pretty low resolution so far. So one thing I can tell you, for example, and this has been published before, is naive CD4 cells are very hard to kill compared to central memory or effector memory cells. The more a cell moves down the differentiation path, the easier it becomes to kill. So if you see a cell that's phenotypically naive, it's going to be harder to kill. But we don't have the data yet to say, are there other markers such as CD155 that are already present on these populations that make them resistant.
And the last question,
Yeah.
BCL2 antagonist is going to affect also the CD8s, the NK cells, who is going to kill these cells?
Yeah. So I think that's a problem. So in our in vitro studies, the CD8 T cell clones that we use aren't as sensitive to BCL2 antagonist as the CD4s. They're more reliant on BCL-XL, but I don't know what it's going to be like in vivo. One project that we have in our lab is a nanoemulsion targeting project to try to target the BCL2 antagonist to the CD4 cells to try to counteract. So I think it'll be important to see the studies of BCL2 as a free drug, especially because there's so much clinical experience with it. But we may ultimately need to think of more targeted approaches for it.
Thank you.
Thank you.
There is another
Yes.
question on the back.
Yes, thank you for the talk. You mentioned in the figure for Matthias' work, they are cells that express RNA, no? So my question is, do you think that, or have you some evidence that in cells infected without expressing RNA, what we can call deep latency, there is also an expression of BCL2 of CD155 markers? I mean, in other words, do we need replication even at very low level of the virus to elicit this phenotype?
Yeah. Yeah, it's another great question. I don't know if we have the technology to parse it out yet. We would almost need something like that. Well, with PRIP-seq, they can identify the RNA positive cells. So has he? I don't know. He must be looking at that and parsing it into cells that are transcribing RNA or not. But I guess I don't know the answer to that.
Okay, thank you.
Yeah.
Brilliant talk, thanks.
Thank you.
Two questions. What is the proportion of these CTL-resistant cells, and when do they appear in the body? Since the beginning of the infection or later on?
Yeah, so a lot of
For
really...
strategical eradication studies is very important.
Yes, so a lot of really good questions. These are basically all the things that are on the frontier that we're looking at. So in terms of the proportion, all that I can say from our in vitro assays is it's about 5% of CD4 cells, but it does vary by clone for reasons that we don't fully understand. So, I would say kind of single-digit percentages, but what does that actually look like in vivo, in the blood, let alone in the tissues? There's a lot of work to be done to figure that out. And how early, if there is selection for an HIV-resistant reservoir, how early does that happen after initiation of treatment? We don't know the answer to that right now. I can tell you how we're trying to explore it is we're taking CD4 cells from people before starting therapy and at multiple time points and pulsing them all with a CMV peptide and trying to kill them all with a clone. And our hypothesis is that as we look later out, doing that will enrich for the infected cells because they'll be more and more resistant. But we don't have the data on that yet.
Thank you.
Thank you.
Great presentation, Brad. Two questions as well. One is, I assume that all you showed today is happening in peripheral blood. So you have any data on what might be happening in tissues, whether the frequency of CTL-resistant CD4 T cells may be higher or lower?
Yeah, a great question. I don't have any data on that. We of course recognize that it's an important area and kind of a blind spot of what we looked at so far. So we'd love to find collaborators to do those studies. We don't have many tissue samples, but at the moment it's all limited to blood, and it might be very different in different tissues.
Right, okay. And the second point is probably more of a comment, is, I think that your data has ignited all the debate again about looking for reservoir in resting T cells versus total CD4 T cells. And I guess that this is to my mind, this is very definitive data to look more directly into total CD4 T cells rather than resting CD4 T cells. Would you agree on that?
I would agree with you.
Okay.
Yes, I would agree with you.
Thank you.
Okay, so we will move to the next presentation. Thank you, Brad.